Bismillahirrahmanirrahim. Thanks for choosing my channel for your learning. This video is about the action potential in the neuron. This video will also discuss refractory period of neuron and propagation of the action potential. Let's begin. Please recall that changes in the resting membrane potential are used as signals in the neurons. These changes occur in the form of either the graded potentials or action potential. If you want to watch our video on the graded potential, the link of that video is available in the description. Action potential is a sequence of rapid and large alterations in the resting membrane potential that is propagated over the entire axon. Action potential serves as electrical signal over a long distance without losing strength. In a neuron, an action potential is also called a nerve impulse. To study the phases of an action potential, we plot a graph of voltage measured across the cell membrane against time. The phases of an action potential begin with depolarization, followed by repolarization. The repolarization goes past the resting potential into hyperpolarization, and finally the membrane returns to rest. These phases of the action potential are related to specific changes in the membrane voltage. At rest, the membrane voltage is minus 70 millivolts. The membrane begins to depolarize when an external stimulus is applied. The membrane voltage has a rapid rise during depolarization phase and reaches to plus 30 millivolts. During repolarization, the membrane voltage starts to return to a negative value. Repolarization continues past the resting membrane voltage, resulting in hyperpolarization. The membrane voltage returns to the resting value shortly after hyperpolarization. Before we discuss what happens during different phases of action potential, we need to look at the stimulus which alters the resting membrane potential to start an action potential. The stimulus is usually in the form of a depolarizing graded potential which begins to depolarize the membrane. Action potential starts when this depolarization reaches a certain level termed the threshold, which is about minus 55 millivolt in many neurons. Threshold in a particular neuron usually is constant. The generation of an action potential depends on whether a particular stimulus is able to bring the membrane potential to threshold. A stimulus which is strong enough to depolarize the membrane from minus 70 to minus 55 millivolts is called as the threshold stimulus. Any stimulus that does not depolarize the membrane potential to minus 55 millivolts or higher will not reach threshold and thus will not result in an action potential. It is called a subthreshold stimulus. A stimulus that is strong enough to depolarize the membrane well above the threshold is called a suprathreshold stimulus. Once an action potential is generated, the amplitude of an action potential is always the same and does not depend on stimulus intensity. Indeed, the greater the stimulus strength above threshold, the greater the frequency of the action potential. This means that several action potentials will form in response to a suprathreshold stimulus, but each of these action potential has the same amplitude as a nerve impulse caused by a threshold stimulus. As you have just learned, an action potential is generated in response to a threshold stimulus but does not form when there is a suprathreshold stimulus. In other words, an action potential either occurs completely or it does not occur at all. This characteristic of an action potential is known as the all or none principle. The all or none principle of the action potential is similar to pushing the first domino in a long row of standing dominoes. When the push on the first domino is strong enough, that domino falls against the second domino and the entire row topples. Stronger pushes on the first domino produce the identical effect, that is, toppling of the entire row. Thus, pushing on the first domino produces an all or none event. The domino all fall or none fall. When a depolarizing graded potential or some other stimulus causes the membrane of the axon to depolarize to threshold, voltage gated sodium ion channels open rapidly. Both the electrical and the chemical gradients favor inward movement of sodium ions, and the resulting influx of sodium ion causes the depolarizing phase of the action potential. As more channels open, sodium inflow increases, the membrane depolarizes further, and more sodium ion channels open. This is an example of a positive feedback mechanism. The inflow of sodium ions changes the membrane potential from minus 55 millivolts to plus 30 millivolts. At the peak of the action potential, the inside of the membrane is 30 millivolts more positive than the outside. In addition to opening voltage gated sodium ion channels, a threshold level depolarization also opens voltage gated potassium ion channels. Because the voltage gated potassium ion channels open more slowly, their opening occurs at about the same time the depolarizing phase is coming to an end. The slower opening of the voltage gated potassium ion channels and the closing of previously opened voltage gated sodium ion channels produce the repolarizing phase of the nerve impulse. During repolarization, the inflow of sodium ions slows down. At the same time, the outflow of potassium ion accelerates. Acceleration of potassium ion outflow causes the membrane potential to change from plus 30 millivolts to minus 70 millivolts. During the phase of hyperpolarization, the voltage-gated potassium ion channels remain open and the membrane potential becomes even more negative to about minus 90 millivolts. As the voltage-gated potassium channels close, the membrane potential returns to the resting level of minus 70 millivolt. The return to the resting membrane potential is also because of the ongoing activity of the leak channels and the sodium potassium pump. While an action potential is in progress, another one cannot be initiated. This effect is referred to as the refractory period. There are two phases of the refractory period: the absolute refractory period and the relative refractory period. During the absolute refractory period, another action potential will never start regardless of strength of the stimulus. This is because the voltage gated sodium ion channel is still in its operational configuration. A new action potential could be started only when voltage gated sodium channel is back to its resting conformation. During relative refractory period, another action potential can be started but only by a stronger stimulus than the one that initiated the current action potential. This is because of the flow of potassium ions out of the cell. Because potassium is rushing out, any sodium ion that tries to enter will not depolarize the cell but will only keep the cell from hyperpolarizing. Action potential always travel from the trigger zone of the axon to the axon terminals. In contrast to the graded potential, an action potential is not decremental. That is, it does not die out. Instead, an action potential keeps its strength as it spreads along the membrane. Thus, the action potential travels along the membrane rather like the activity of that long row of dominoes. This mode of conduction is called propagation. Propagation of action potential happens in two ways. These are continuous conduction and saltatory conduction. Continuous conduction involves step-by-step depolarization and repolarization of each adjacent segment of the plasma membrane. In continuous conduction, ions flow through their voltage-gated channels in each adjacent segment of the membrane. In actuality, it is not the same action potential that propagates along the entire axon. Instead, the action potential regenerates over and over at adjacent regions of the membrane from the trigger zone to the axon terminals. In a neuron, an action potential can propagate in this direction only and cannot propagate back towards the cell body. Because any region of the membrane that has just undergone an action potential is temporarily in the absolute refractory period and cannot generate another action potential. Note that the action potential propagates only a relatively short distance in a few milliseconds. Also, continuous conduction occurs in unmyelinated axons. Saltatory conduction is the special mode of action potential propagation that occurs along myelinated axons. there is an uneven distribution of voltage-gated channels in the myelinated axons. Only few voltage-gated channels are present in regions where a myelin sheath covers the axolemma. By contrast, at the nodes of Ranvier, where there is no myelin sheath, the axolemma has many voltage-gated channels. Hence, current carried by sodium ions and potassium ions flows across the membrane mainly at the nodes. When an action potential propagates along a myelinated axon, ionic currents are generated at the first node. This serves to depolarize the membrane to threshold, opening voltage-gated sodium ion channels at the second node. The resulting ionic flow through the opened channels constitutes a nerve impulse at the second node. Then, the action potential at the second node generates an ionic current that opens voltage-gated sodium ion channels at the third node, and so on. Later, each node repolarizes after its depolarization. The flow of current across the membrane only at the myelin sheath gap has two consequences. Firstly, the action potential appears to leap from node to node as each nodal area depolarizes to threshold. When an action potential leaps across the long segments of the myelinated axolemma, it travels much faster than it would in an unmyelinated axon of the same diameter. Secondly, opening a smaller number of channels only at the nodes represents a more energy efficient mode of conduction. Because only small regions of the membrane depolarize and repolarize, minimum inflow of sodium ions and outflow of potassium ions occurs each time a nerve impulse passes by. Thus, less number of ATP are used by sodium potassium pumps to maintain the low intracellular concentration of sodium ions and the low extracellular concentration of potassium ions. The speed of propagation of an action potential is affected by three major factors: amount of myelination, axon diameter, and the temperature. As you have just learned, action potentials propagate more rapidly along myelinated axons than along unmyelinated axons. Larger diameter axons propagate action potentials faster than smaller ones due to their larger surface areas. Cooling of the axons causes propagation of action potential at lower speeds. Thanks for watching and listening. Do share this video with your friends and colleagues. If you like this video, subscribe to this channel for watching more videos.